Hello. To get started, I'd like to share that I began this work as an employee at the Florida Fish and Wildlife Conservation Commission, FWC, but I now work for the Biological Resources Division of the National Park Service. I'm extremely grateful for the continued collaborations with FWC and that my current supervisor, Jenny Powers, allowed me to follow this project through completion. Aside from my co-authors, Andrea Sylvia and Brittany Bankovich, I'd like to acknowledge the many others at FWC who assisted in this work, as well as Brian Stacy at NOAA Fisheries and numerous students and employees at the University of Florida's Veterinary School. Finally, I'd like to put a plug in for Dr. Becky Hardman's talk on Tuesday morning. She is a veterinarian at FWC, and she will have some updated information regarding field investigations and surveillance. Approximately 52% of 360 species of turtles and tortoises are categorized by the IUCN as threatened with extinction. Two of the top reasons for population declines are habitat loss and overexploitation for human consumption and the pet trade. Other contributing causes of mortality include vehicular collisions, predation, and disease. In today's presentation, I will be discussing turtle fraservirus1, or TFV1, an emerging disease of freshwater turtles in the southeastern United States. The disease was initially observed in March 2018 in Florida softshell turtles within the St. Johns River system, which flows from south to north on the eastern side of Florida's peninsula. Given the numerous threats facing turtle populations, it is very important to increase awareness of novel diseases such as TFV1. My presentation is divided into two parts. I will first summarize some background information and then delve into results from an epidemiological analysis. The initial field investigations, TFV1 discovery and characterization, and the retrospective finding of a nearly identical case in a farmed softshell turtle were described in 2022 in PLOS Pathogens. Phylogenetic analyses of two viral proteins revealed that the virus formed a distinct clade under the phylum Nagarnivericata and belonged to a new viral family, which we named Tosoviridae, after the Tosohatchee Wildlife Management Area where the index cases occurred. Further taxonomic assessment is ongoing. Since its initial discovery, this RNA virus has been detected in multiple species of freshwater turtles in the Florida Peninsula. Some turtles may test positive for TFV1 without demonstrating clear clinical signs of disease. However, many infected turtles will be obviously ill. Sick turtles might lack strong defensive behaviors. They can be slow to flee, easy to capture, and may not attempt to bite. They might be found either partially submerged or completely out of the water, with their necks extended and held flat to the ground. Other signs of illness include abnormal breeding and skin lesions, as well as emaciation in some species. The information presented on this slide comes from 93 turtles that were necropsied between March 2018 and September 2021. 47 of these turtles were positive for TFV1. The most common necropsy observation in positive turtles was the presence of exudative plaques in either or both the oral cavity and the cloacal region. However, it should be noted that this is a nonspecific lesion, which can also be seen with other diseases, such as rhinovirus. Positive softshells were generally in good body condition with adequate adipose tissue. In contrast, positive cooters were more likely to be emaciated. For both groups of turtles, stomachs were usually empty. Adult females were often in a gravid condition or in a state of vitellogenesis with abundant yolk formation. Diagnosis of TFV1 infection can be confirmed via molecular methods and histopathology. Conventional and quantitative reverse transcriptase PCR methods have been developed. This seems to be a systemic virus, and PCR can be performed on a variety of sample types. Cloacal and oral swabs, as well as urine samples, can be tested from live animals. Multiple tissues can be tested from deceased animals, including kidney, brain, liver, and spleen. In situ hybridization demonstrates virus presence across many tissue types with a high degree of staining, especially apparent in renal tissue. Across species, a consistent finding on histopathology is vascular-associated inflammation within the nervous system. There are many unknowns regarding the epidemiology of TFV1. For example, how is the virus transmitted? How many turtle species are susceptible hosts? Does infection always result in fatality, or can some turtles be non-clinical carriers? Is the virus distribution geographically restricted based on climate or other factors? What long-term population impacts might be expected as a result of this emerging disease? Without these answers in place, FWC leadership took a proactive approach and enacted Executive Order Number 21-19, which includes prohibiting the take and transport of all softshell turtle species in Florida. Prior to enacting this order, anyone could capture and transport a softshell turtle outside of the peak nesting period. With emerging diseases, when there are so many unknowns, it can be difficult to design disease surveillance strategies and formulate management guidelines. Therefore, in the absence of formal pathogen challenge studies, we set out to evaluate the available data to inform preliminary guidance. To begin to address some of these questions, we evaluated TFV1 infection status and metadata in an epidemiological framework. We created 4 separate logistic regression model data sets for 2 reasons. First, we needed to accommodate our sample size of 93 animals. Second, we wanted to analyze tiers of available data, which may vary depending on financial and personnel resources. For each model, the dependent variable was the TFV1 infection status based on PCR results. The field model used data that could be collected from public citizens reporting mortality events and field investigations. The necropsy model contained data obtained from experienced personnel who performed necropsies in a laboratory setting. The weather model primarily pulled data from 2 online resources. Precipitation data came from the Community Collaborative Rain, Hail, and Snow Network, or COCORAS, and temperature data came from the National Weather Service. We also included day length, which was calculated using the R package INSOL. In today's talk, I will not further discuss the weather model dataset. Finally, the spatial model used GIS-derived data. I won't have time today to fully explain each variable and the justification for its inclusion in the analyses. Instead, I will just highlight the significant findings. The field model dataset included 4 categorical variables. First, we assigned turtles to a species group as either a softshell or hardshell. Second, we categorized turtles as having been collected in either the wet or dry season. Third, based on landscape analyses within a 1-kilometer buffer surrounding the turtle, an urbanicity score of low, medium, or high was given. And finally, we evaluated the morbidity and mortality simple cluster status, which we termed CL7. A yes value was assigned if more than 1 sick or dead turtle of any species was reported in a given location within a 7-day period. Otherwise, the turtle received a no for this variable. In this figure, the predicted probability of a turtle testing positive is on the y-axis. As shown, the CL7 variable was highly significant. Based on the estimated odds ratio, turtles were 18 times more likely to test positive for TFV1 if they were collected from a CL7 event. The necropsy model dataset included 5 variables. The first 4 were species, sex, maturity, and plastron length. The 5th variable was termed any plaques. Turtles were assigned a yes value if they had plaques located in the oral cavity, cloacal region, or both locations. The interaction between any plaques and species was significant, which you will see as I walk you through this figure. The red lines represent the soft shells and the black lines are the hard shells. Pairwise comparisons are significantly different when the codes A and B above each line cannot be matched across pairs. So, for example, on the left side of the graph, there is no difference in the probability between soft shells and hard shells testing positive when neither of them have plaques. Both lines have an A code. In contrast, if we compare soft shells without plaques, code A, to those that had plaques, code B, a large difference is seen. We designed the spatial model to evaluate spatiotemporal patterns beyond the conservative CL7 approach that I described in the field model. We created least cost paths between every possible pair of turtles, prioritizing movement through water. We then set 4 mutually exclusive cluster distances, which I show in this target schematic. The red star in the center represents the location of a negative or positive turtle. The orange circle represents a tight cluster, which contains all positive turtles within a 1 kilometer distance of the starred turtle. The purple circle represents a loose cluster, which contains all positive turtles between 1.1 kilometers to 5 kilometers from the starred turtle. The cluster distances were based on published linear home ranges of freshwater turtle species. For each additional positive turtle within the tight cluster, the starred turtle was approximately 4 times as likely to test positive. For each additional positive turtle within the loose cluster, the starred turtle was approximately twice as likely to test positive. When we add in a time component of plus or minus 30 days, the probabilities of the starred turtle testing positive increase slightly. The southeastern United States is one of the richest areas in the world for turtle biodiversity, and Florida is among the top states in freshwater turtle species. The potential population impacts of TFV1 to softshell turtles and other freshwater turtles is currently unknown. Many turtle species do not produce offspring until they are 10 years or older, and only a few of these offspring will survive to become reproductive adults. Therefore, the loss of mature individuals can have significant effects on turtle populations. It was such concerning to find deceased, very large female softshells that were gravid or in a vitellogenic state. Furthermore, infected turtles were often found with comorbidities such as bacterial or fungal pneumonia. It is possible that these were secondary infections or that the turtles were more susceptible to TFV1 because of these infections. I'll conclude with a few summary points on management and surveillance options. Our characterization of cases as clustered mortality events is compatible with a highly transmissible waterborne virus that is shed in urine or other secretions. Management options should include biosecurity practices such as the implementation of published disinfection protocols, restricting or limiting human-assisted turtle movement among locations, and 1 or more PCR screening tests of turtles admitted to wildlife rehabilitation centers prior to their release from these centers. Surveillance strategies should be developed based on TFV1 detection goals and resources. For example, the finding of oral or cloacal plaques in softshell turtles, especially if the animal came from a clustered mortality event, can be suggestive of TFV1 infection. In this situation, molecular testing might not be done to conserve financial resources. Field investigations and testing could be prioritized to hardshells and new species or by geographic locations. And with that, I'd like to thank you for your attention. I'll be happy to address questions now if there is time or later in the conference.